Hello students, today's our topic is DNA replication. DNA replication is very crucial process in the cell cycle. It helps in the transfer of information from one generation of cells to another. The very important point to note here is DNA template on which replication takes place is in the direction 5' to 3', and the direction of replication is 5' to 3'. So this must be remembered by you when we study the replication process in detail. So we know that DNA is a double-stranded polynucleotide where the two strands are anti-parallel to each other. That is, one strand is running from 3' to 5' direction and another strand is running from 5' to 3' direction. They are having hydrogen bonds in between the nucleotides over here, and in turn they are joined here by diphosphoester bonds over here. Okay, so in between you have the hydrogen bonds and they are anti-parallel in direction. One strand is running from 3' to 5' direction and another is running from 5' to 3' direction. The DNA template on which replication takes place is always in 3' to 5' direction, and the direction of replication is always in 5' to 3' direction. Okay, now let's look into detailed steps of replication. The first step here is initiation. The initiation or the beginning of the DNA replication occurs at the specific sites on DNA molecules called as origin of replication. In E. coli, these sites are called as oric. Okay, at this sites itself the replication begins. These are the specific sites. At these sites, enzyme helicase first comes and binds to the DNA molecule. Okay, this enzyme helps in separation of double-stranded DNA into single-stranded DNA. By which enzyme? By enzyme helicase. It separates the double-stranded DNA into the single-stranded DNA. The second step of replication is helicase action. Helicase is an enzyme which is initiating the unwinding process at origin of replication. At origin of replication, helicase enzyme is initiating the unwinding process. So suppose if this is the double stranded DNA, when helicase enzyme comes and binds at the origin of replication, it starts separating the double stranded DNA into the single stranded DNA by breaking these hydrogen bonds. Okay, so here the double stranded DNA is converted into single stranded DNA. Okay, now the third is, once the DNA is converted into the single stranded DNA, some proteins comes and binds over here to the single stranded DNA. These proteins are called as SSB proteins. Now what are these SSB proteins? They are single stranded binding proteins. These proteins prevent the single stranded DNA from forming the secondary structure among itself, or prevents them from re-annealing, that is rebounding, or prevents secondary structure formation. Okay, so SSB protein comes in, joins here. This is helicase, which is breaking the hydrogen bonds in single stranded DNA. So when helicase is breaking the double stranded DNA into the single strand DNA, here generally, because of the rotation or something, tension is created ahead of the replication fork. Now this is the replication fork which is formed. So ahead of replication fork, there is always tension created by the helicase enzyme, okay, in the double stranded DNA. So to remove this tension, here another enzyme comes and binds, that is topoisomerase. So our fourth step is binding of topoisomerase. Topoisomerase comes and binds ahead of the replication fork, that is ahead of even helicase enzyme. What does it do? Topoisomerase relieves the tension generated by unwinding of DNA strands. By creating temporary breaks in the strand and again resealing it. Okay, so this was our fourth step. Till here what have we learned? We have learned that helicase first comes and binds at the origin of replication. It separates the double stranded into single stranded DNA by breaking the hydrogen bonds, and once this single stranded DNA is formed, SSB proteins comes and binds to the single stranded DNA so that the further rebounding or re-annealing is prevented. Apart from this, our second enzyme topoisomerase comes and binds ahead of the replication fork or ahead of even the helicase enzyme, so that it relieves the tension generated by the unwinding of the DNA strands by causing temporary breaks and then again resealing it. Okay. Now the replication fork has been formed. Once the replication fork has been formed, This is our topoisomerase. I'll write it as topo. Then these are our SSBs. Okay. Now this is our replication fork. Once our replication fork is formed, our fifth step is primer synthesis. Here RNA primers helps in attaching RNA primer. So which enzyme is acting now? RNA primase synthesizes RNA primer. This RNA primer is complementary to the single stranded DNA template, and it acts as a starting point for the DNA synthesis. We know that DNA is antiparallel in direction. The double strands are antiparallel in direction. So now this is our 5 prime to 3 prime and 3 prime to 5 prime. Okay. Now our sixth step is that binding of DNA polymerase. Okay. Therefore, here after RNA primer is synthesized and attached to the complementary DNA strand, our DNA polymerase 3 comes and binds over here. Okay, it starts to attach nucleotides. It attaches nucleotides in 5 prime to 3 prime direction. The template is in 3 prime to 5 prime direction. The DNA polymerase attaches nucleotides into 3 prime to 5 prime direction. So in 5 prime to 3 prime direction, DNA polymerase 3 attaches nucleotides and its replication or the synthesis is done. Now here it is 3 prime, but here it is 5 prime. We know that DNA templates must always be in 3 prime to 5 prime direction. So here the primer comes and attaches on the RNA primer comes and attaches on the lagging strand. Here only once the RNA primer attaches, here multiple times RNA primer attaches on the lagging strand. So this is the leading strand. Now let me explain this in detail over here. We have helicase here, we have topoisomerase, we have SSBs, and we have one RNA primer here, and this is 3' to 5' direction, and this is 5' to 3'. We know that the template always must be 3' to the 5' direction. So this template is in 3' to the 5' direction, but this template is in 5' to 3'. So when the template is 3' to 5', it is called as leading strand, and when it is 5' to 3', it is called as lagging strand. Okay. Now once RNA primer is attached, synthesize the RNA primer, the DNA polymerase 3 comes and attaches to this RNA primer and started attaching nucleotides from 5 prime to 3 prime direction. This direction of DNA primase is fixed. It will always synthesize in 3' to 5' direction. But on lagging strand, that is our 6th step, what will happen on lagging strand is that it is in 5' to 3' direction. So the DNA polymerase 3 cannot synthesize in 3' to 5' direction. The template must be from 3 prime to 5 prime direction only. Therefore, RNA primer attaches primase attaches RNA primer at different locations over here, and here the DNA polymerase 3 comes and attaches, and it synthesizes in 3' to 5' direction only. Okay, and these are called as these small fragments. It is continuously synthesizing, but here it is synthesizing in small fragments. Okay, these small fragments are called as Okazaki fragments. Okazaki fragments. Now let's see here about the formation of Okazaki fragments. They are formed on the lagging strand. Many such fragments are formed as many RNA primers attach themselves on the lagging strand. On leading strand, it is a continuous synthesis because the DNA polymerase is synthesizing in 3 prime to 5 prime direction, and it is continuous because the template is in 5 prime to 3 prime direction. Okay, so here many Okazaki fragments are formed. Okay, the primers as many RNA primers are on lagging strand. Therefore, many starting points which results in many Okazaki fragments. Okay. Now, DNA polymerase 3 extends these RNA primers on the lagging and leading strand both. DNA polymerase 1 removes the RNA primers once extension is done and replaces them with DNA nucleotides. Okay, once extension is done, they are replaced. DNA polymerase 1 comes, removes the RNA primer and replaces it with the DNA nucleotides. Now, this is a replication fork. We have a helicase, we have a topoisomerase, we have SSB, we have RNA primer here, we have DNA polymerase 3. Now, and synthesis is occurring over here. Here we have small Okazaki fragments happening. This is leading strand, this is lagging strand. Now when small Okazaki, this is again RNA primer here, RNA primer here, RNA primer here, and here polymerase is extending this. So now what happened, once this RNA polymerase 3 comes nearer to this RNA primer, DNA polymer 1 comes and removes this RNA primer and attaches the nucleotides. Okay, this RNA polymer primer is replaced with DNA nucleotides, okay, by the action of DNA polymerase 1. Now here the joining must be done between these fragments, which is done by ligase action. Okay, so the ligase, the ninth step is ligase action. Ligase seals the gaps between the Okazaki fragments on lagging strand, creating a continuous double-stranded DNA on lagging strand. Okay, all the Okazaki fragments are sealed with the help of the ligase enzyme. Next comes our last step. Here is proof reading and repair. DNA polymerase have proofreading ability. Our DNA polymerase enzyme 3, especially 3, has the proofreading ability. It corrects proofreading ability. In the sense, it corrects its mistakes. In mismatching nucleotides. Okay, mismatch mechanisms further correct errors that may have escaped this proofreading ability of DNA polymerase 3, which we will be dealing in our next class. Okay, the result of DNA replication is 2 identical double-stranded DNA molecules. So now let's complete the entire figure of DNA replication. Here we have the helicase, we have topoisomerase, we have SSBs here, okay, 3' to 5', 5' to 3'. Okay, so 5' to 3' is our lagging strand. So here our Okazaki fragments are made. Okazaki fragments, and from 5' to 3' R is a leading strand. There is continuous synthesis by DNA polymerase 3, which is having our 5' to 3' synthesis, Here, because the template is 5' to 3', it has become the lagging strand, and here the template is 3' to 5', it has become the leading strand. Okazaki fragments are formed. The sealing is done by ligase. The removal of primer is done. RNA primer removal is done by DNA polymerase 1. Once the primer is removed, the DNA ligase seals the gap. So thus the replication of the DNA, where two strands are synthesized. Okay, here also once the sealing is done, the strands are synthesized. Double stranded DNA are synthesized on two single stranded DNA templates. Okay student, in next class we will see the replication errors.